This call is now being recorded.
चलिए आते हैं हमारे यहाँ पे गैमेटोजेनेसिस के ऊपर. I think female reproductive part is very clear to you. We have done the external genitalia. If you remember, we have done also along with that we have done the main reproductive part. That's our ovaries, oviduct, uterus, vagina, cervical canal. All the terms we have covered, and you know what kind of question they can ask you from those particular terms. So the thing which we have started with yesterday was my gametogenesis, जहाँ पे हम बात कर रहे थे हमारे यहाँ पे spermatogenesis के बारे में. Here we were discussing something in terms of the spermatogenesis. So here you will get to see that initially, as I've told you, you have a large number of cells which are there in the seminiferous tubules, and those cells over there are nothing but my particular spermatogonia. So the cells which we get to see over there is going to be spermatogonia, something we have seen that. Now these spermatogonia are my diploid cells, right? And which are coming from my male germ cells. You can also call them as directly as a male germ cell. So male germ cells are also called as my spermatogonia. They will undergo mitosis. They are going to increase their number. So now you have a large number of diploid cells. This large number of diploid cells will slightly grow in size. And now you are going to call them as a primary spermatocyte. So primary spermatocyte is also diploid in nature. याद रखना अभी तक मैंने यहाँ पे meiosis start नहीं किया है. I haven't started with the meiosis over here. Now what will happen? My primary spermatocyte will undergo meiosis 1 and will give me 2 cells. And these 2 cells are called as my secondary spermatocytes. Okay. So primary spermatocyte undergoes a meiosis 1 and gives you the 2 cells that are my secondary spermatocyte. And the secondary spermatocyte will undergo the meiosis 2, and then you will have a formation of your 4 spermatid. So that is how you will be finding your 4 spermatid are being formed. तो ये होता है इस तरीके से आपके spermatid यहाँ पे आपके बनेंगे. Please remember very carefully where the meiosis is going to start. It always starts from the primary spermatocyte. ठीक है? Spermatogonia से meiosis start नहीं करना है. Okay, यहाँ से start करना है. So यहाँ से आपने start किया, and this is what you will get. You will get in the end 4 spermatid are been formed. Now once you have the spermatid has been formed, they don't have a well-defined shape. इनके पास कोई well-defined shape नहीं होता. They do not have any kind of well-defined shape. और क्योंकि इनके पास कोई well-defined shape नहीं होता, you have to give them a well-defined shape. You have to give them a look which appears like a sperm, right? तो conical head, लंबी सी tail, वो आपको देना होगा यहाँ पे. तो आप यहाँ पे क्या करते हो? So this is something we have done. Now what you will do? You will convert this spermatid into your spermatozoa. So now this spermatid, which are like round structures over here, they will now have a appropriate kind of shape, and that is called as my spermatozoa. And this process over here is called as my spermiogenesis. The process which will be taking place over here is going to be called as my spermiogenesis. So they will ask you, what is your spermiogenesis is known for? तो बताना आप clearly कि spermiogenesis का मतलब होता है कि आपका spermatid जो आपका यहाँ पे होता है, that will convert into your spermatozoa. That will ultimately convert into your spermatozoa. So is this clear to all of you? क्या ये चीज आप सबको यहाँ पे clear है?
Yes, sir.
अच्छा, you cannot see, yes, you still cannot give me the reactions, ना? I think तनिष कभी भी वो ऑप्शन नहीं आ रहा क्या रिएक्शन देने का? नहीं आ रहा ना? रुको, let me fix that. Let me quickly fix that. Send reactions. Chat message तो वैसे भी तुम लोग नहीं करते, so I think उसका तो कोई मसला है नहीं. रिएक्शन आप देख पाओगे. इससे मुझे रिलीफ मिलता है कि हाँ जी समझ आ रहा है, नहीं आ रहा. थम्स डाउन भी दे सकते हो अगर समझ नहीं आ रहा हो तो. ठीक है. So ये हमने यहाँ पे देख लिया. So we have seen what has been happening with your spermatozoa, कैसे आपके यहाँ पे बन रहा है. अब ये आपके यहाँ पे बन चुका है. Now the spermatozoa has been formed. Now let's see what will be happening. Now this spermatozoa will get its head embedded in the Sertoli cell. ये जो मेरा spermatozoa मेरा यहाँ पे होगा, the spermatozoa which will be present over there, this spermatozoa will have its head inside the Sertoli cell. तो इसका आपका head आपको लगा हुआ यहाँ पे लगा हुआ inside the Sertoli cell. So it will embed the head inside the Sertoli cell. Yeah, so ये मेरा यहाँ पे Sertoli cell. And this is the male gamete, right? So sperm head inside Sertoli cells. So this is something you have to know. So your sperm head will be embedded inside the Sertoli cell. So आपका उसका सर वहां पे लग जाता है Sertoli cell के अंदर. So now your basically spermatozoa are been formed. Now the next thing you find happening here is, अब इसके बाद मेरा अगला स्टेप यहाँ पे क्या होगा? So remember one thing, sperms have been formed in my seminiferous tubule. Can anyone tell me where the sperms undertake maturation? या maturation यहाँ पे कहाँ पे होता है? Where is the location where you find the sperm undergo maturation or they attain the motility? कौन सा लोकेशन था जहाँ पे मोटिलिटी आ रहा था, मैच्योरेशन आ रहा था? बताओ. तनेश, शिखा, मंताशा, व्हाट वाज द लोकेशन जहाँ पे आपका स्पर्म का मैच्योरिटी और आपका मोटिलिटी आता था मेल रिप्रोडक्टिव पार्ट के अंदर?
Epididymis.
राइट, एपिडाइडाइमस. कुछ याद आ रहा है, नहीं आ रहा? सब इवेपोरेट हो गया. हाँ. Epididymis is going to be a structure जहाँ पे आपका sperm का motility और sperm का maturation होता है. So there is a location where we will finding the sperm motility and the sperm maturation will take place. Now to reach over here, the sperm has to go out of the this seminiferous tubule, right? So now understand what will happen. ठीक है, ये तो हमने साइड में लिख दिया कि आपको रिवाइज कराने के लिए कि यहाँ पे आपका क्या होता है. This is the location where we will be finding the sperm undergo maturation and the sperm attain the motility. Okay. Now the thing which you have to remember here is that once the sperm has been formed, once the spermatozoa has been formed, now from here it will be released out into the cavity of the seminiferous tubule. तो यहाँ से इसको release किया जाएगा. So now the sperm will be released from this particular site. So it will be released from the seminiferous tubule. So ये इसको हम यहाँ से release कर देंगे. So now what will be happening? My sperm will be released from the seminiferous tubule. Be released from seminiferous tubule. So basically अब यहाँ पे क्या होगा, that these sperms which have been there present and getting formed in the seminiferous tubule will be released from that particular site. So they are released from that particular area so that they can move on to the next area. They can move on to the next destination, which will eventually will be initially ready testes आएगा, then we'll have a vas efferentia, and then we'll find getting into the epididymis. So that is something you have to understand, that now the spermatozoa once has been formed, they'll be released from the seminiferous tubule. And when they are released from the seminiferous tubule, this event, releasing of spermatozoa from the seminiferous tubule, this event is called as my spermiation. This event over here is called as my spermiation. So this event of releasing of my sperms or spermatozoa from the seminiferous tubule, and now they'll be sent into the lumen, right? From the tubule they'll be sent into the free space. इनको आप यहाँ से निकाल के free space में आप भेजोगे, फिर वहां से you can send them into the your particular epididymis तक आप उन्हें भेजना चालू करोगे. But this release of this spermatozoa is called as my spermiation. तो हमेशा याद रखना what do you mean by spermiation and what do you mean by spermiogenesis. ये दोनों बहुत ही closely related आपके यहाँ पे terms होते हैं. You should be very clear what they actually mean, right? When someone says spermiogenesis, then you have to know that your sperm is getting, your spermatid is getting converted into your spermatozoa. That is called as my spermiogenesis. And when someone talks about spermiation, that is nothing but my release of sperm from the seminiferous tubule. ये सबसे सिंपल तरीका याद रखने का कि यहाँ से वो निकल रहे हैं, यहाँ से वो निकल रहे और एक बार वो यहाँ से निकलेंगे तभी तो आगे जाएंगे, तभी तो at that time only they will go into the epididymis. गीतांजलि is there in गायत्री is there in the class or not? गायत्री हो आप क्लास में? हाँ बेटा टाइम पे आने का, टाइम पे. गायत्री is be on 3:30. ठीक है. हाँ. So that is something you will be finding happening over there. You will find there will be release, and that is called as spermiation. So this should be clear to all of you. ये clear होना चाहिए सबको. शिखा, तनिष्क, गायत्री and Mantasha. I hope it's clear to all of you. ठीक है. So now we can see again the picture over of my spermatogenesis. You can see the meiosis 1 happens, then you'll find the meiosis 2 will happen, and that is your spermatid, right? So that's something we have seen. Spermatid are transformed to spermatozoa by a process of my spermiogenesis, something we have already discussed. So this is something we are all familiar with. Okay, so this is what you find happening, something we have seen in the last class. Spermiogenesis, sperm head becomes embedded in the Sertoli cell, something I've just told you, they become embedded in the Sertoli cell and are finally released from the सेमिनिफरस ट्यूब्यूल्स, और बाद में इनको हम रिलीज कर देते हैं सेमिनिफरस ट्यूब्यूल्स में से, एंड दैट इवेंट ओवर हियर will be called as my spermiation. This event is called as my spermiation. So please remember these terms very carefully, right? So now there's one thing you have to be very much clear with over here, that when does the spermatogenesis begin? So something I've told you, that spermatogenesis is going to initiate
At puberty.
Yes, right. As soon as the boy is going to hit the puberty, that's when the spermatogenesis is going to be is going to initiate. Now there's one thing you have to understand. So as soon as a boy is going to hit the puberty, there are few things you have to understand over here. That first of all, there will be a release of some hormones from the hypothalamus. So you'll have the hypothalamus, and from the hypothalamus, some hormones will be released. From the hypothalamus, certain hormones are released. So basically, the hormone coming from the hypothalamus is going to be your GnRH. GnRH. Gonadotropin releasing hormone. You call them as, ये आपने पढ़ा होगा 11th में, chapter 21. Gonadotropin, last chapter. Gonadotropin releasing hormone, right? Now, what is GnRH does? ये GnRH जो आपका आ रहा है hypothalamus में से, ये जाके बोलेगा आपके pituitary gland को. It will go and tell the anterior pituitary. It will go and tell the anterior pituitary gland. It will go and tell the anterior pituitary gland to start releasing two hormones. One is going to be called as your FSH, follicular stimulating hormone, and other is called as my LH. So these again names we have seen in the 11th grade. So I'm expecting that you know these names, right? So you have these two names over here. You have FSH and you have LH, right? So ये दो आपके यहाँ पे जो दो नाम हैं, ये आपको मिलेंगे, ये आपके आ रहे from the anterior pituitary. So these are my two hormones: follicle stimulating hormone and luteinizing hormone. So these two hormones are coming from the anterior part of my pituitary gland. Okay. Now these two hormones actually go and actually promote the formation of my spermatogenesis. So these two hormones are the one which actually trigger or promote the formation of your sperm, and sperm formation are going to be promoted by them, right? Like I can give you some example, like you have your FSH, sorry, you have your LH. LH is known to go and tell the Leydig cells. LH is known to go and tell the Leydig cells to start releasing one hormone. And which hormone is going to release? Leydig cell से कौन सा hormone आएगा? अरे क्या दोपहर को आके बैठे? Yeah, it is going to be a testosterone. Leydig cells क्या release करते बाबा? Leydig cells are going to release testosterone. शिखा याद आ रहा है? शिखा, गायत्री, मंताशा. Leydig cells are going to release something like your testosterone, which will be coming from the, which will be coming under the influence of my LH. So LH जाके बोलता है, LH will go and tell the Leydig cells to release the testosterone. So LH will go and tell the Leydig cells to release the testosterone. So that is something which will be coming from the Leydig cells, right? So FSH will primarily play the role in promoting the spermatogenesis. So FSH primarily plays a role in promoting the spermatogenesis. Now let's have a look at few things. So spermatogenesis starts at the age of a puberty due to significant increase in the secretion of my gonadotropin releasing hormone. Something we have discussed, right? From hypothalamus. The increased level of GnRH then act on the adenohypophysis. Adenohypophysis means anterior part of my pituitary gland, right? And stimulates the secretion of two type of hormones: luteinizing hormone LH and my follicle stimulating hormone. So these two hormones are basically coming out from the anterior part of my pituitary gland. ये सब याद रखना है, बाद में मैं आपसे पूछूंगा, ये सब याद होना चाहिए. Okay. Then what will be happening after that? Once you find happening, now let's have a look over here. LH act on the Leydig cells and stimulate the secretion of androgen. So as I've told you, LH will go and act on the Leydig cells और वहां से क्या करेगा? आपका androgen बनाने में आपके में मदद करेगा. Okay. And then you have an androgen in turn stimulate the process of my spermatogenesis. So जैसे आपका androgen बना, androgen मतलब testosterone. Please घबराना मत या मिक्स मत करना इस चीज को कि एंड्रोजन ही आपका यहाँ पे टेस्टोस्टेरोन है, ठीक है? So that androgen is something which is going to stimulate the process of your spermatogenesis. So this is where you will find the process of spermatogenesis is going to start. Okay. Now what happens after that? FSH act on the Sertoli cells and stimulate the secretion of some factor which help in the process of my spermiogenesis. So FSH जो आपका है यहाँ पे, it is actually promoting predominantly in the process. Again, it is specifically helping to carry out the again spermatogenesis only. But यहाँ पे उसका specific काम क्या है? It will go and act on the Sertoli cell. It will go and act on the Sertoli cell, and then you will find this Sertoli cell are the one which actually release some factors. ये जो Sertoli cell आपके यहाँ पे है ना, सुनो मेरी बात ध्यान से. FSH आके क्या बोलेगा? It will go and tell the Sertoli cells. It will go and tell the Sertoli cells to stimulate some factors, right? Secretion of some factors. Now, what are those factors? So these factors are called as my androgen binding protein. So yaad rakhna, the factors which are coming from the Sertoli cell, they are called as my ABP, which stands for your androgen binding protein. So that is something coming from the Sertoli cell, and this ABP actually helps in the process of a spermiogenesis. That actually helps in making the spermatozoa from the spermatid. So this is one thing you have to know. There's something you have to remember over here. That is what they are known for. So इतनी बात यहाँ पे याद रखना, इतनी चीज याद रखना कि LH कहाँ पे क्या काम कर रहा है. Or you can directly remember like this, that LH will go and act on the Leydig cells. LH will go and act on the Leydig cells. On the other hand, my FSH will act on the Sertoli cells. And collectively, both of them are making sure that you are having spermatogenesis will takes place. Okay. So this story, even though it's a very simple story, you have to remember this thing. Exam में कितना भी वो लोग घुमा के आपसे पूछे, आपको इसका आंसर आना चाहिए. You shouldn't be have any concerns with solving this. ठीक है. So that is something you have to know about the spermatogenesis. सबको समझ आ रहा है यहाँ पे? मंताशा, गायत्री, तनेश, शिखा, I hope it's clear to all of you.
Yes, sir.
कोई कंसर्न आए तो बताना. ठीक है. ओके, चलो. और रिपीट करना हो तो भी बता देना. So that is something we have done. Now let's have a look at the spermatozoa. Now let's have a look at the sperm, how the sperm is going to look like, and what are the traits I'll get to see in that. Right? So this is how the sperm is going to look like. Now sperm is going to have like three parts. It will have, or I will say the body will be divided into three parts. We'll have the head part. We'll have a middle region. You have a middle part. And you have a tail. These are the three regions in which you basically find the sperm is getting divided into. Now let's try to talk about these regions over here. Now when I discuss something like a head part of the sperm is going to be having two important things over here. First of all, it will be having the haploid nucleus. It will have the haploid nucleus. You know that now all the sperms are going to be haploid in nature, so this will be having the presence of a haploid nucleus will be seen in them. So that's one thing you have to know. It will have the haploid nucleus. That's one thing. Second thing, what you get to see, the tip of the head. The tip of the head, this conical part over here. The tip of the head, this conical part over here is called as my acrosome. Is going to be called as my acrosome. So this head part is going to have something known as something like your acrosome. Now, acrosome is well known for having a large number of enzymes. You get to see a number of enzymes in the acrosomes. So, acrosomes are going to have a number of enzymes, and you can also have enzymes. You can either use a term like enzymes over here, or you can also use a term like lysins. Right? Now, what this acrosome is going to have, or what kind of enzyme is going to have? So, enzymes or lysins which you have in the acrosome, they are used for penetration of the egg. जब आपका sperm, when the sperm travels towards the egg will have the outer covering. The egg will have the outer covering, and that outer covering has to be broken down, or that outer covering has to be penetrated. So outer covering has to be digested, and that can be done with the help of this acrosome. So acrosome is at the tip of the sperm, right? This is like a group bag of enzymes. हाँ, इसको तुम ऐसे भी याद रख सकते हो. It's a bag of enzymes. एक नहीं होते, बहुत सारे होते. It's going to be having it's like a bag of enzymes. It's a bag of enzymes. And these bag of enzymes will have one role to be played. They will actually help the sperm to penetrate the egg. It actually helps in the penetration of the sperm. It helps in sperm penetration. Penetration into egg. That should be clear to you that is what my acrosome is known for. Okay. That is something you have to be very much clear with. So that's what you find in the head. You have a nucleus which is going to be a haploid nucleus. यहाँ पे आपका DNA भी होगा, chromosomes भी आपके यहाँ पे होंगे, and then you have the head part which will be having the acrosomes. Okay, acrosome is an important part. Acrosome is made up of Golgi apparatus. Golgi, what is going to make this acrosome? Please write it down somewhere. And this will have the enzymes, lysins, and they are going to penetrate your particular head. Penetrate the egg ka membrane. Okay. Then you have the middle part. Now, middle piece or middle part is going to be having your mitochondria. So, middle part is something you can see over here, and this will be having the mitochondria. Now, as the mitochondria is there, you can very clearly understand what kind of role it will play. This mitochondria will play a role in providing the energy. इसका एक ही काम यहाँ पे होगा, to provide. It will play a role in providing energy. So, energy provide करने में ये आपकी यहाँ पे करेगा मदद. So that is one thing you have to know, that mitochondria will be seen inside the center part over here, and that mitochondria will have a role to play in providing the energy. ठीक है, तो यहाँ से आपकी energy आती है. So that's the middle part. And then you have the tail. Then you have an elongated tail over here. There's an elongated tail over here. So tail is nothing but something which will be used for the motility. So you have this tail, and I hope it's very clear that tail is something which can be used for the motility. ट्रैवल करने के लिए. तो यहाँ पे जो आपकी एनर्जी बन रही होती है ना, जो आप माइटोकॉन्ड्रिया से एनर्जी बना रहे होते हो, एनर्जी मींस व्हाट? एटीपी. माइटोकॉन्ड्रिया इज नोन फॉर व्हाट? मेकिंग एटीपीज. दोस एटीपीज विल यूज फॉर द मूविंग दिस टेल. सो दिस टेल एक्चुअली हेल्प्स इन द मोटिलिटी. जितना फटाफट टेल मूव करेगा उतना जल्दी मेल गैमेट आगे बढ़ेगा. द मोर फास्टर द टेल इज गोइंग टू मूव, द मोर फास्टर द मेल गैमेट इज गोइंग टू मूव टुवर्ड्स द fallopian tube. So energy has been used to generate this movement of the tail, and that is what the tail is known for. Okay. Now there are two things you have to remember over here. You have a neck region. You can see a small neck region over here. You have a neck region, and the neck region will be having presence of a centriole. Neck region will have a presence of a centriole. Now there is one thing you have to be very much clear with, that centrioles are over here going to be of my two types. So I'll discuss the centrioles. So I have centrioles. Let me discuss centrioles later on in the next slide. But right now, initially remember that you have a head, you have a middle piece, you have a tail. Middle piece is known for mitochondria, presence and generate energy, and head is known for having haploid nucleus and acrosome, which actually helps penetrating the egg. क्या इतनी बात यहाँ पे समझ आ रही है? Is this thing clear to all of you?
Yes, sir.
All right. Okay. All right. चलो ठीक है. So you can see over here the neck, you can have a plasma, you can have a tail, you can see all those things over here. Now what actually happens now? In the neck you will have a, let me just see, you will have a presence of a centriole. Now let me just write down two things. You have a, in the neck region you have a presence of a centriole. You know that, right? And that centriole in the neck region is called as my proximal centriole. The centriole which you have in the neck region is called as your proximal centriole. Proximal centriole. Now, what this proximal centriole is known for? This proximal centriole is basically to help during the cell division. Now, you know that you need to have a spindle fiber formation during the process of a cell division. So you always have to form spindle fiber. अगर आपको याद हो cell division आपने पढ़ा है. So whenever someone has to undertake the cell division, there is the need to form the spindle fiber, right? Now, once in future, if the egg fertilize the if the sperm fertilize the egg, then there will be a requirement of cell division. After fertilization, there will be a requirement of cell division, and for cell division, you will be needing a centriole, and that centriole will be seen over here. So याद रखना, जो neck में आपको यहाँ पे centriole मिल रहा है, that centriole is called as my proximal centriole, and that centriole is actually the one which plays an important role during the cell division after fertilization. If sperm successfully reaches that particular egg, then if possible, if the fertilization takes place, then after that you have to undertake the cell division. तो उसके लिए ये यूज किया जाता है. तो भूलना मत, एक बहुत important point मैंने आपको यहाँ पे बताया. You have to remember this things, that what is my centriole over here is going to be known for, right? So please write it down, this point, that centriole is known to promote cell division after fertilization. Okay, so that is something you can do over here. So that is something you have to know, right? Then you will also have one more centriole which is not been shown over here, and that centriole something can be seen over here. एक और centriole आपको मिलेगा somewhere over here, and that centriole is called as my distal centriole. That centriole is called as my distal centriole. That centriole is called as my distal centriole. Now, distal centriole is basically used for formation of the flagella, tail. ठीक है, इसमें यार इतना याद रखना है कि यहाँ पे जो centriole था ना, उसे हमने use किया to form this tail. So you had one centriole over there which has been used to form this particular tail. If you just remember, if you just go back in your chapter of your cell the unit of life, we have discussed this, that centrioles are going to form the basal body, and from the basal body you are going to form the flagella. किसी को याद आ रही है, हमने बात किया था जो लोग ब्रिज कोर्स करते हैं, right? तनिष्क को याद आ रहा है, बाकी शिखा आई होप आपको भी याद आ रहा होगा, right? Gayatri, Mantasha, that you have the centrioles which converts into basal body and then into the flagella. So that is something you find happening over there. So that's why distal centriole is being used for forming this particular tail, which is nothing but my flagella. So that is something you have to know. Okay, so these are two couple of points you have to remember. Now, so these are the things we have discussed. Let's have a look at them. The head of the spermatozoon contains an elongated haploid nucleus. We know that. The head is covered by a cap-like structure called as my acrosome. That's a bag of enzymes. That's going to be a bag of enzymes, right? Acrosome contains numerous enzymes like protease, acid phosphatase, hyaluronidase, right? Remember the names of enzyme at least these three names you have to remember. That helps in the fertilization of the ovum. So basically helps in the penetration of the ovum, right? Then the middle piece possesses numerous mitochondria which produces energy for the movement of the tail. So you have a middle piece which produces a number of mitochondria which will be used making energy for the movement of the tail. Sperm motility is essential for fertilization. You need to have motility, and then only you can find the fertilization can take place. So that much amount of information should be very particularly clear to all of you, right? That's what you'll be finding happening over here, right? You can have a look at the picture of the NCERT. NCERT picture आप यहाँ पे देख सकते हो. You can very clearly see the picture of NCERT over here, right? Human male ejaculates around 200 to 300 million sperm during coitus. Now these are the numbers which you have to remember. These are the numbers of the NCERT, and these are the numbers जिसको आपको याद रखना है. These numbers have to be remembered by any means. So human male ejaculates around 200 to 300 million sperms during the coitus. And you should know that how much amount of sperm should be having the motility. At least 60% of the sperm must have normal shape and size, and at least 40% of them must show vigorous motility. So around 60% of the sperm should have normal shape and size, and at least 40% of them should have a good amount of motility. Then only you can find the normal fertility will be seen, and then only you can see there's a possibility of getting conceived. There's a possibility of attending the pregnancy in a simple terms. So that is something you have to know. So ये basic requirement you need to have in the sperms. Okay. Now this is something, it's something which you have to remember. इसमें कोई बहुत ज़्यादा understanding नहीं है. There is nothing which has to be understood over here. You have to remember all those things. Now, also, if you remember, we have discussed it that when the sperms are being released, sperms are not the only thing which is getting out of the penis. You will be finding you have a seminal plasma. There's going to be a seminal plasma. I hope you remember the seminal plasma, the secretion which was coming from the three gland. What were the names of the three glands? शिखा बताओ क्या था नाम उन तीन gland का जहां से ये plasma आता था? शिखा बताइए. सी 4 लोग की क्लास में तो एक को पढ़ के क्वेश्चन पूछा जाएगा. इसकी आदत डाल लो आप. शिखा इतना टाइम. अरे वो ग्लैंड्स कौन से थे बेटा? ग्लैंड्स, वो तो 3 ग्लैंड्स कौन से जहां से आपका सेमिनल फ्लूइड आता था? टेल अस गायत्री, टेल अस which are the 3 glands.
So seminal vesicle, prostate gland, bulbousine gland, ureteral gland.
Right. So these are the 3 glands from which you'll be finding the secretion of seminal plasma is going to come from them. Right. So that is something you have to know. There's something I've already discussed that you will have a seminal plasma, and seminal plasma plus sperms is called as, ये दोनों को मिला के हम क्या बोलते हैं? What is being called as? वैसे ही बता दो अनम्यूट करके. Seminal plasma sperm is called as what? You people are not, क्या कर रहे हो तुम लोग, पढ़ाई कर रहे हो नहीं कर रहे? हाँ जी सीमन, कितना टाइम लग रहा है आने के लिए आंसर? इतना कैसे टाइम लगा सकते हो आप लोग? So semen, sperms plus seminal plasma is called as semen, right? Now please remember, whatever you are finding in the seminal plasma is going to nourish the sperm. Something we have already discussed that, right? We have already discussed that. I won't be getting into detail of that. You know that seminal plasma will have number of things, and that is going to nourish the sperms, right? Nourish the sperm, make the movement of the sperm smoothly. It will ensure that the passage of the urethra is not acidic. So everything is being taken care by the seminal plasma. So something you know that, right? So I won't be getting into detail. I won't be repeating those things again. ठीक है, ये हम already कर चुके हैं, right? Now, so this is where the story of the sperm is going to end. यहाँ पे sperm की story आपकी यहाँ पे होती है खत्म, and now we're going to start with a new topic, that is your oogenesis. So here we are going to initiate one of the most important, I would say important to सभी है, but one other topic which has some details which has to be understood, that is going to be the oogenesis. So चलिए, let's start with the oogenesis and let's try to understand the story of the oogenesis over here. ठीक है? So I hope सबको यहाँ तक समझ आ रहा है? By the way, the things we have covered, I hope it's clear to all of you. यहाँ तक Gayatri, Tanish, Shikha and Mantasha, I hope it's clear to all of you. यहाँ तक
Yes, sir.
right. Now let's start with the oogenesis. Now let's start with the story of the oogenesis and let's try to see what will be happening over here. Now oogenesis simply means what? Again, it's a very simple thing. Now whatever I'm going to tell you, just try to follow everything step by step. अगर कुछ समझ नहीं आता, then पूछ लो, बीच में ही पूछ लो, रोक के पूछ लो कि सर ये समझ नहीं आया, because there are number of things which has to be understood over here. So that's why just pay attention. Okay. So now when we discuss oogenesis, the first thing you know, it is nothing but my formation of ova. The simple terms, formation of ova in ovary. Formation of ova in ovary is called as my oogenesis, right? That's what you'll finding happening over here. Now, the process of oogenesis initiates at the embryonic stage. This initiates at embryonic stage. At embryonic stage. So now at the embryonic stage only, you will find the oogenesis in the female is going to start. So embryonic stage, we can also call it a fetal stage. Embryonic stage आप इसे बोल सकते हो, or you can also call it as a fetal stage, right? So when the baby is actually developing inside the mother's body, at that time only you will find the oogenesis is going to initiate. Now what happens during oogenesis? You will find around 2 million oogonia are being formed in the ovary. Around 2 million oogonia are formed. Now this is all happening when the female child hasn't even born, right? Now understand this thing, when the oogonia are formed, oogonia is something coming from the germ cells. And like spermatogonia, oogonia is also going to be deployed in nature. So around 2 million oogonia are being formed during the fetal stage, and they are also going to be deployed in nature. Okay. Now please understand, apparently the birth has not taken place. The birth has not taken place. Now what happens now? Some of the oogonia over here, like a large number of oogonia, what will happen? So they have been formed over here and they have been formed via mitosis. So they are been getting formed. When I say they are been formed over there, how they are been formed? They are getting formed via mitosis. So कैसे वो बनाए जा रहे हैं यहाँ पे? They are been formed via mitosis. So through mitosis they are getting formed. So पहले ऐसे 4-5 से ऐसे, फिर उसके बाद बहुत ज़्यादा नंबर बढ़ जाता है. You find a number getting increased via mitosis, right? So that will be happening over here. Now what happens? That you have a large number of oogonia in the fetal stage. Now, some of the oogonia, right? Some of the oogonia, or I would say large number of oogonia, will initiate the meiosis 1. Or I won't say like this, right? I will rather say like this, that you have your, assume that this is a oogonia, right? This is a oogonia, diploid cell. Okay. It will initiate the meiosis 1, right? It will initiate the meiosis 1. तो ये start करेगा अपने यहाँ पे meiosis 1. It will initiate the meiosis 1. Now understand one thing, it has initiated the meiosis 1, not completed. It will initiate the meiosis 1 and will get arrested at the prophase 1. After initiating the meiosis 1, it will get arrested at the prophase 1. तो meiosis उसने चालू कर दिया. Let me try to show you in a different way. Forget this. इसको हम ऐसे बताते हैं, थोड़ा और आसान करते हैं इसको. यहाँ मेरा oogonia था, इसने इनिशिएट किया आपका यहाँ पे meiosis 1. It has initiated the meiosis 1. So meiosis 1 over here is going to start. But will it get completed? The answer is no. So meiosis 1 you have started, but you have not completed. So it will get arrested. It will get arrested at prophase 1. At prophase 1. So now what you will get to see that your oogonia have initiated meiosis 1, but it has not gone beyond that. It has not gone beyond that. It has arrested at a prophase 1 पे आके वो रुक गया. And now this oogonia will be called as my primary oocyte. So now when you have a oogonia which has initiated the meiosis 1 and will stop at prophase 1, that oogonia over here is called as my primary oocyte. So that oogonia here is called as my primary oocyte. So if they ask you what you get to see in a primary oocyte, if you take out the, if you see the ovary of a newly born baby, or I would say if you see the ovary of a baby which is still developing in the mother's body, then you can find a large number of primary oocytes can be seen. And if they ask you that these primary oocytes are in which particular stage, so you have to tell them that they have started the meiosis 1, but they have got themselves arrested in the prophase 1. So they have not completed the meiosis 1. याद रखना meiosis 1 चालू किया, complete नहीं किया. अगर complete करेंगे तो आपको 2 cell मिलेंगे. If you complete the meiosis 1, you will get a 2 cell. Apparently they have just started the meiosis 1 and they have arrested themselves. So you will find a large number of oocytes will be like this, and they are called as my primary oocyte. Okay, so that is one thing we have to know. So what will happen now? At the time of a birth, when the baby is born, so let me just show you, explain you over here. At the time of a birth, at birth, you have around millions of primary oocytes. Millions of primary oocytes are present. Primary oocytes are present. And when I say this primary oocytes are going to be present, so please remember this thing, they are the one which are like in a arrested state. They are arrested as a prophase 1. They are going to be present over there, right? Now, there's one thing you have to understand here is that from birth till the girl attains puberty, a large number of oocytes are going to die. So they are going to remain inactive, they are going to remain in an arrested condition. And from birth till the girl attains a puberty. So from birth till puberty, what will you get to see? That only 60,000 to 80,000 primary oocytes remains, or I would say survives. Remaining all degenerate. So when a girl hits a puberty, let's say after birth, after 13 years, when you observe in the ovary, you will get to see that this much number of oocytes are being seen in the ovary. Itne hi number of oocytes you will find in the ovary, and so that means a large number of oocytes have died. That itself means that a large number of oocyte have undergone death, and that is something you find happening. That is a very normal thing you will find. Anyway, this is the number you will be finding in at the time of puberty, and these are the number of oocytes she'll be using throughout her lifespan. So these are the number of oocytes she'll be using throughout her lifespan. So that should be clear to all of you. तो इतनी बात याद रखना कि प्राइमरी oocyte आपके जो इनिशियल स्टेज पे थे at birth, and whatever you find by the end of the girl hits puberty is going to be way more less. A less number of primary oocyte will be there at a time of puberty, right? So that's the first thing you understand over here. Now the second thing you have to understand here is that within the ovary, when the primary oocyte is formed, let's say the baby is still developing inside the mother's body. Let's say the baby is still developing inside the mother's body. So what will be happening? Acha, one more thing, no more new oocyte will be formed. सुनो मेरी बात ध्यान से, वापस coming back. Whatever the oogonia is formed initially, ना उसके बाद addition नहीं होता. Whatever the oogonia has been formed initially, after that no more new oogonia is been added over here. So don't expect that. Like in males, what happens? The sperms are keep on forming. The new your spermatogonia just keeps on forming. You don't find that happening in oogenesis. Whatever has been formed during the fetal development and after birth, you don't find any kind of other growth happening. You don't find any other new oogonia getting added over here. जितना है उतना ही रहेगा वो. कम होगा but ज़्यादा add नहीं होगा. You won't find getting increased over there. Number getting increased. Okay. Now understand one thing carefully. So you have a primary oocyte which is in the meiosis 1, prophase 1. It has been arrested over there, right? Now it is not alone actually. So assume that this is my primary oocyte. Assume that this is my primary oocyte. So primary oocyte is surrounded by a group of cells. Primary oocyte is surrounded by a group of cells. So you'll have a layer of cells surrounding the primary oocyte. A layer of cells surrounding the primary oocyte. And this layer of cells surrounding the primary oocyte is called as granulosa cells. Granulosa cells. So now you have a single layer of granulosa cells surrounding the primary oocyte, and in the center you have your primary oocyte. So let me mark this as well. This is my primary oocyte. Primary oocyte. And this structure over here is called as my primary follicle. So you have an outer layer, outer covering of single layer of granulosa cell, and that is over here can be called as my primary follicle. So this structure over here is called as your primary follicle. This is called as your primary follicle. I won't write like this, or else you'll get confused. I will write it like the primary follicle, the primary follicle. So this structure over here is called as your primary. Okay. So assume that what will happen at a time of a birth, the female child will be having around a large number of oocytes will be there inside the ovary, and all they have a covering of this granulosa cells. All of them will have a covering of a granulosa cells, and that is called as a primary follicle. So always the primary oocyte is within the primary follicle. And now assume that this is the girl who has just born right now, and after 13 years something will happen over here. So first of all, you have to know कि birth के time पे आपको देखने के लिए आपको यहाँ पे मिल जाए. So you have a primary follicle and inside you have a primary oocyte. Okay. Now first of all, tell me यहाँ तक समझ आ रहा है नहीं आ रहा? Is this thing clear to you यहाँ तक सबको? Okay, गायत्री, तनिष्क आ रहा है समझे नहीं आ रहा? तनिष्क बताओ. चलिए मैं रिपीट कर रहा हूँ. हाँ, मैं रिपीट करता हूँ. See what will be happening? इसके अंदर देखो यहाँ पे हमने क्या देखा that you have formation of like oogenesis is called as my formation of ova. This will start at the embryonic stage. At a fetal stage, it will start. So baby develop कर रहा है तो आपका ये start हो जाएगा. Around 2 million oogonia are been formed. Around 2 million oogonia are been formed via mitosis. Right? Oogonia are always going to be deployed in nature. And now these oogonias, a large number of oogonia, what will happen? They will initiate the meiosis, but will stop at the prophase 1. They will get arrested at a prophase 1. मतलब meiosis 1 मेरा खत्म नहीं हुआ. चालू हो गया लेकिन खत्म नहीं हुआ. So they will get themselves arrested at a prophase 1, and that oogonia can be called as my primary oocyte. That oogonia will be called as my primary oocyte. Okay. Now what happens? So now you have a primary oocyte. This primary oocyte, which has been there, now assume that the baby has born. So once the baby has born, within the ovary, you will have millions of primary oocyte. Within the ovary, you will have millions of primary oocyte will be seen in the at birth. But as the girl hits puberty at the age of 13, now if you do the counting of the primary oocyte, the primary oocyte are going to be less. The number of primary oocyte which are remaining at a time of puberty is going to be around 60 to 80,000. Okay. Now understand what will happen after this. So now you have, so this is something you have to know. Now after this, you have to know one more thing, that the primary oocyte which was there in the baby or which was there in the fetal stage was not alone. It had a covering. It had an outer covering. उसके आसपास outer covering थी. और जो outer covering उसके आसपास थी उसे हम बोलते हैं यहाँ पे as a granulosa cells. So granulosa cell के एक outer covering आपको यहाँ पे देखने के लिए मिल जाती है और सेंटर में आपका होता है आपका primary oocyte. और इस स्ट्रक्चर को हम बोलते हैं यहाँ पे एज अ प्राइमरी फॉलिकल. दिस स्ट्रक्चर ओवर हियर इज कॉल्ड एज प्राइमरी फॉलिकल. सो नाउ अज्यूम दैट, सो दिस इज अ कंडीशन. सो वंस अ गर्ल hits puberty, a large number of primary follicle are there inside the ovary. Okay, now understand what will happen. समझ आया यहां तक? तनिष्क समझ आया?
यस सर.
ठीक है, चलो और बढ़ते हैं. Now what will be happening, that you have this primary follicle. Now carefully understand what I'm going to tell you now. So ये कहाँ से आ गया? Let me just एक सेकंड, 2 मिनट रुको. Just get rid of this. All right. Okay. So that's our primary follicle. Now let's discuss something in terms of the primary follicle in detail. So now let me show you over here itself. So you have a primary follicle, and as soon as a girl hits puberty, so as soon as a girl is going to hit puberty, so assume that a girl hits a puberty over here. So at puberty, we'll find certain things are going to change. What is going to change? There's going to increase the number of hormones in the body, right? So under the hormonal influence at puberty, under hormonal influence, under the influence of hormone, which hormone, how hormone, something we'll see that later, but under the influence of the hormone, what will happen? The primary follicle will develop. The primary follicle is going to develop. So this is something should be clear to all of you. At puberty, under hormonal influence, what will happen? The primary follicle is going to develop. So primary follicles develops. Primary follicle develops. Now let's see what actually happens with the primary follicle. Let's try to see that how the development is going to be happening over here. So now let's pay attention and let's try to do some kind of development over here. So in the center, you have your primary oocyte. You have the outer layer over here. Outer layer of your granulosa cells. That was my primary follicle, right? Now what happens with this? Now it will undergo some kind of development. So it will change. So what kind of changes I will find over here? Suddenly now you have a thick outer layer. You have a single layer of granulosa cells. Now suddenly you will be having a much more thicker layer of granulosa cells will be seen over there. So let me just draw a. Right. So now I'll make a slightly thicker layer of your granulosa cells. Okay. So assume that you have multiple layers of granulosa cells will be seen over here. Multiple layers of granulosa cells will be seen. And in the center, you have the oocyte. In the center, will be having the oocyte. So now let me label this again. So you have your primary oocyte. You have the granulosa cell. And this is going to be called as my primary follicle. This was your primary follicle. My primary follicle. Now what happens over here? Now you have primary follicle has been converted into the secondary follicle. It has been converted into your secondary follicle. Now secondary follicle is something in which you will find a multi-layers of granulosa cell will be seen. So over here the layers of granulosa cell will be much more higher. So multi-layered granulosa cell have been observed over here. So you can very clearly see you have a multi-layered granulosa cells are seen. Such cells have been observed. So एक लेयर नहीं होती, आपको मल्टीपल लेयर आपको यहाँ पे मिल जाएगी. So मल्टी लेयर्स ऑफ granulosa cells will be observed over there. तो काफी ज्यादा आपके यहाँ पे बन जाते हैं. पहले एक ही थे, अब यहाँ पे काफी ज्यादा बन चुके हैं. Right. So that's one thing you will get to see over here. Along with that, the oocyte remains in the center. Oocyte will always remain in the center. That's going to be seen. So that is the difference you can observe. So now suddenly you have a large number of cells which can be seen in my secondary follicle. So you can see a large number of cells can be seen in my secondary follicle. Okay. Now what has been seen that after that there is a formation of a theca. So आपको क्या मिल रहा है यहाँ पे? You have a multi-layer of granulosa cell, and you also have one type of cell which you can see over here, and that is called my theca. There is also one more type of cell which you can see over here, and those cells are called as my theca. So one more type of cell can be observed over here, that is called as my theca. So we'll have something like your, all right. So you have some theca cells also. Just draw some random cells just to tell you. Call them the theca cells. Now this picture which I'm drawing over just to make you understand few things. Huh? They have been drawn to make you understand few things. That you have to draw some cells and you have to remember those cells. So some cells have been seen over here, which are called as my theca. So theca is also observed. Okay, and this theca which I am observing over here, it will have like two parts. It will have the inner and outer theca. It will have the inner and outer theca. You can see over there, right? Theca interna. So it will have two parts. You will have something like your theca interna. You have theca interna. And there will be a presence of a theca externa. Theca externa. Right? No one is going to ask you in way too much detail of this type of in detail, but you have to know few things because they can become part of a statement. So you will have a theca over here. Theca are nothing but a kind of a cell, and those are into my two parts. You have a theca interna, you have a theca externa. Okay, so that is something you have to know. Now after this, you have to know these are my secondary follicle. Now what will happen after the secondary follicle? You will find there will be a formation of my tertiary follicle. So from here, there will be a formation of my tertiary follicle. Now, before understanding tertiary follicle, there's one thing you have to remember. There's one thing you have to remember all the time that, chalo ye mera hai, yahan pe mera, like we have seen the secondary follicle has been formed. It will have a multi-layer of granulosa cell. It will have a secondary, it will have a theca, and that is called my secondary follicle. Okay. There's one thing you have to understand here is that your oocyte, which you have in the center. So you have a oocyte in the center, primary oocyte, and it is covered by a layer. It is covered by a layer. So this layer has to be remembered. It is covered by a layer, and that layer is called as my zona pellucida. So my oocyte is been covered by a layer, and that layer is called as my zona pellucida. तो ये बात याद रखना कि आपका जो oocyte, आपका जो प्राइमरी oocyte है, that will be covered by a layer which is called as my zona pellucida. So that should be clear to all of you. This is a layer I'll be finding in the primary oocyte all the time. Okay, so that is the one layer. So if they ask me ये layer क्या है, cell है या क्या है, तो याद रखना zona pellucida is nothing but it's a glycoprotein. It is nothing but your glycoprotein. That's it. ये आपका यहाँ पे glycoprotein है. Okay. इसके अलावा बाकी जो यहाँ पे cells है, that's going to remain the same. So you can again draw your all your particular cells. We'll get to see. You can see them, right? Now what happens now? Now understand this thing very clearly. You have your primary oocyte in the center, right? You have all your granular cells which have multiple layers, and you also have a presence of a theca. Along with that, you also have a presence of a theca, right? So I'll just make some random cells just to show you. You also have a theca over here. Some random cells over here. Yeah. अब आपको यहाँ पे एक बात यहाँ पे याद रखनी है. There is one thing you all have to remember here carefully is going to be that your secondary follicle has been formed. This will now convert into my tertiary follicle. This will now convert into my tertiary follicle. Now, as soon as it's converted into tertiary follicle, there are two things you have to understand. First of all, in a tertiary follicle, you will have a large fluid-filled chamber can be observed. Tertiary follicle के अंदर आपको एक बड़ा सा fluid-filled chamber आपको मिलेगा. You will find in tertiary follicle, you will have a large fluid-filled chamber will be observed, and that fluid-filled chamber is called as my antrum. That fluid-filled chamber is called as my antrum. So you should know that in the tertiary follicle, this is my tertiary follicle. This is my tertiary follicle. And this tertiary follicle will have a fluid-filled chamber. It will have a fluid-filled chamber. And this fluid-filled chamber is called as my antrum. So you can see the antrum over here. Theca तो आपको यहाँ पे है, बन चुके पहले से. आप यहाँ पे you have a primary oocyte, and primary oocyte is been surrounded by my zona pellucida, right? It will have a surrounding of your zona pellucida. That is something you have to know, right? Now, as soon as the tertiary follicle has been formed, now one thing you have to very clearly understand that my primary oocyte will now complete my meiosis 1. मेरा जो प्राइमरी oocyte मेरा जो अंदर है ना यहाँ पे, अब वो यहाँ पे मेरा meiosis 1 कंप्लीट करेगा. ये बात आपको याद रखनी है कि जो मेरा प्राइमरी oocyte है यहाँ पे, now this will convert to meiosis 1. अभी तक meiosis 1 इसने कंप्लीट किया था या नहीं किया था? अरे बोलो, इसने meiosis 1 किया था नहीं किया था? नहीं किया था इसने. अभी तक meiosis 1 इसने कंप्लीट नहीं किया था. कौन से स्टेज पे अटका हुआ था ये?
प्रोफेस 1.
Right, it was अटका हुआ था in the prophase 1 के अंदर वो अटका हुआ था. So याद रखना, as soon as the tertiary follicle has been formed, what will be seen, one of the key trait is going to be that now primary oocyte will complete the meiosis 1. Primary oocyte will complete the meiosis 1. Okay. Now understand this thing very carefully. As soon as the meiosis 1 gets completed, now you will find a secondary oocyte has been formed. So now my primary oocyte will now complete the meiosis 1, and now let's see what happens. See, याद रखना हमेशा. अब मैं आपको यहाँ पे थोड़ा इजी तरीके से बताता हूँ. So you should know this thing very carefully. ये समझो इस बात को. इसको हम इसको यहाँ पे डिवाइड कर देते हैं. You had your primary oocyte. ठीक है? You had your primary oocyte over here. Okay, this was my primary oocyte, and now it has completed my meiosis 1. So now it has completed the meiosis 1. Now, as soon as it has completed meiosis 1, I will get two cells. मुझे 2 सेल यहाँ पे मिलेंगे. So I'll be finding my 2 cells will be formed, and the 2 cell will be of my unequal length. The 2 cells which have been formed will be of my unequal length. So I'll have one very large cell. That one large cell which I'll be forming over here, that one large cell over here will be called as my secondary oocyte. That will be called as my secondary oocyte. And there will be formation of one small cell as well. There is one very small cell which will be formed, and there's one small cell which will be formed over here. That will be called as your polar body. Polar body. So after meiosis 1, you will find 2 things will be formed. You will find a secondary oocyte will be formed and a small polar body will be formed. So 2 cells have been formed of an unequal size. 2 cells of unequal size are in form. So unequal size cells can be observed. So unequal size cells are observed. ये आपको यहाँ पे देखने के लिए मिल जाएगा, right? So that is the outcome of my meiosis 1. So now my secondary oocyte is been formed. So now you have a tertiary follicle, and the tertiary follicle now will be having the secondary oocyte. So as soon as the tertiary follicle is getting formed, you find the primary oocyte will complete the meiosis 1 and will form the secondary oocyte. अब एक बात बताओ, now tell me something. क्या आपका meiosis पूरा खत्म हो चुका है, अभी भी बाकी है?
बाकी है.
meiosis 2
बाकी है जी, बाकी है बिल्कुल. Now you still have primary, you still have meiosis 2 has to be done, right? So now understand what will happen. Secondary oocyte आपका बन चुका है. Secondary oocyte has been formed over there. Now this secondary oocyte will initiate the meiosis 2. Now this secondary oocyte is going to initiate the meiosis. So I'll draw over here. So now you have your secondary oocyte, the larger cell. You have your secondary oocyte over here, and this secondary oocyte will. This is my secondary oocyte, and this secondary oocyte will initiate the meiosis 2. Will initiate. Just wait a minute. Will initiate the meiosis 2, but will get arrested. ये भी रुक जाएगा, but will get arrested. But will get arrested. Now the question is कि कहाँ पे arrest हो रहा है? Why it is getting arrested? Where it is getting arrested? Why and what is the purpose over there? So first of all, you remember that secondary oocyte will initiate the meiosis 2, but will stop in between. तो ये कहाँ पे रुकेगा? उसकी भी गाड़ी रुकी थी. That also stop in between. Now this will also stop in. This is also stopping in between. So this is stopping in between your metaphase 2. So it will get arrested at the metaphase 2. So at metaphase 2, this will get arrested. So ये वहां पे आकर रुक जाएगा. ये बोलेगा कि अब मुझे यहाँ पे meiosis आगे नहीं बढ़ाना, right? So now you have what? So now you have a tertiary follicle with the secondary oocyte inside it, and that secondary oocyte is arrested at the metaphase 2. So मेरा metaphase 2 चालू हो चुका है, but complete नहीं हुआ है. क्या इतनी बात आप सबको समझ आ रही है? मंताशा, तनिष्क, who else we have? गायत्री, शिखा. तनिष्क बताओ, समझ आ रहा है नहीं आ रहा? तनिष्क joined the class. तनिष्क
yes sir
आ रहा है समझ में यहाँ तक?
yes sir
ठीक है, ये सब याद रखना है, सारा इंपॉर्टेंट है. ठीक है, so that is something you have to know. This is something you should be very much clear with, right? So secondary oocyte will also have a zona pellucida. याद रखना, वो secondary और primary सबके पास zona pellucida होता ही है. Okay, now let's do one thing. You have now your tertiary follicle will convert to graphene follicle. Now tertiary follicle will convert to graphene. So graphene follicle is nothing but a larger follicle. So graphene follicle is nothing but a larger follicle, right? And this larger follicle will have like more number of cells, right? It will have more number of cells now, right? ज़्यादा number of cells यहाँ पे. What was that fluid filled cavity? क्या थे वो fluid filled cavity का नाम?
antrum
Right. So antrum आपको यहाँ पे मिल जाएगा, आपको यहाँ पे देखने के लिए. Antrum will be seen over here, right? So this antrum will be seen, and then you also have a secondary oocyte which has been ready over here. Secondary oocyte has been formed, right? Secondary oocyte which has initiated the meiosis 2 but has arrested at the metaphase 2. That is my secondary oocyte. Okay, these are all your so this large tissue, this large follicle which has been formed over here, this large follicle is going to be called as my Graafian follicle. This large follicle over here is going to be called as my Graafian follicle. Right? It has everything. It has all those theca, it has all those multi-layer of granular cells. Everything will be seen over there. Okay? So this is called as my Graafian follicle. Graafian follicle. It is the most mature follicle. इससे ज़्यादा बड़ा नहीं हो सकता ये. इससे ज़्यादा बड़ा आपको ये होते हुए आपको नहीं मिलेगा. So Graafian follicle is the last follicle which has been formed. Now understand, all this follicle development is all happening with the help of the hormones, right? So because of the hormones, all this development is happening. So as soon as a girl hits a puberty, every month a single primary follicle will get developed into a Graafian follicle. And during that development, you will be finding the primary oocyte will convert into secondary oocyte, and that is what you will be finding happening over here. So this is something should be very much clearly understood, that this is all happens in the initially. So you have initially a primary follicle. As soon as so start from here. As soon as a girl hits a puberty, what will happen? Under the influence of the hormone, your primary follicle will start developing. Right? So every month a single follicle will undergo the development. So my primary follicle, which was having what? Outer layer kya thi? Granulosa cell, and you were having inside, you were having the primary oocyte. And now this primary follicle will now convert into my secondary follicle. Now secondary follicle will have what? It will have the multiple layer of granulosa cell, and will also have a presence of a theca. You will have something like my theca interna, you will have something like your theca externa. That is my secondary follicle. Now this secondary follicle will grow more in size and become my tertiary follicle. Now once the tertiary follicle has been formed, please remember that it will have more number of cells, but it will also have a fluid-filled cavity which can be very clearly seen, and that can be called as my antrum. Now once the tertiary follicle has been formed, this is the time when I'll be finding my primary oocyte will complete my meiosis 1. जो मेरा अटका हुआ था प्रोफेज 1 के अंदर, वो मेरा यहाँ पे meiosis 1 complete करेगा, and the outcome is going to be my secondary oocyte. Outcome is going to be over here is going to be my secondary oocyte. So here the secondary oocyte will be formed, right? So it's something we have seen over here. The primary oocyte will convert into your will complete the meiosis 1, will give you the secondary oocyte, which will be a larger cell, and you'll have a smaller polar body will be seen over here. So now you have two cells. One cell is much more larger in size than the other cell, and so unequal cells have been formed, and that is something you find as an outcome of my meiosis 1. So after the meiosis 1, you have a secondary oocyte. It will also initiate the meiosis 2, but it will get arrested at the metaphase 2. And after the tertiary follicle, एक बार आपका tertiary follicle यहाँ पे बन चुका है ना, after that it will more grow in size, and now it can be called as my Graafian follicle. So Graafian follicle will be having all those large number of granulosa cells, right? So you can see over here, you will have a large number of like granulosa cells over here can be seen. These are all my granulosa cells over here. In the center, you have the antrum. You have a granulosa cells. You will also have presence of your theca. You can also see your antrum can be seen over here. This is the most developed Graafian follicle can be seen. And then you have your secondary oocyte, which has been ready over here and arrested at a metaphase 2. अटका हुआ जो मेरा यहाँ पे है, वो मेरे यहाँ पे खड़ा हुआ है. So this is something you have to know. This is all happening once a girl hits a puberty, and this is all happening under the influence of the hormone. तो पहले तो ये बताओ, क्या इतनी बात समझ आ रही है, नहीं आ रही? सच्ची बताओ. तनिष्क यहाँ तक समझ आ रहा है?
Yes, sir.
Tanish, Mantasha, Gayatri, Shikha, I hope it's clear to all of you. This should be like super clear to all of you that what has been happening over here and why it is being done. Right? Now what will happen? Now understand what will happen. The last step over here is going to be, so the follicle development has been taken place. Now there's one thing I want to explain you separately. There's one thing I want to add over here, that whenever you discuss about the oocyte, whenever you talk about the oocyte, whether it's a primary or it's a secondary, you should know that even if it's a secondary oocyte, you should know it will have an outer layer. A secondary oocyte will have an outer layer. What is that outer layer? Batao. क्या नाम है इसका? गायत्री, व्हाट इज द आउटर लेयर? हाँ, या मंताशा बताओ, व्हाट इज द आउटर लेयर?
Zona pellucida.
Right. The outer layer is also always going to be called as your zona pellucida. So you'll have outer layer. This outer layer is called as my zona pellucida. What is zona pellucida made up of?
Glycoprotein.
Glycoprotein.
Glycoprotein, right? Zona pellucida is made up by glyco. Very good. So that is something we'll get to see. Now, along with zona pellucida, you should know one more thing. So that is something we'll get to see, but you will have one more layer of cell is seen surrounding the oocyte. In the secondary oocyte, you will have one more layer of cell can be seen surrounding them. One more layer of cell can be seen. It is something which comes later. एक बार आपका secondary oocyte बन गया, तो you'll have a layer of cells seen surrounding them. So you have a zona pellucida, तो आपका है ही वहां पे, but एक layer होती है आपकी cell की जो आपको मिलेगी यहाँ पे. And that layer of cell is called as my corona radiata. Corona radiata. So now you have two layers. So now your primary oocyte, your secondary oocyte was having zona pellucida, वो तो था ही, but उसके साथ it has one additional layer can be seen surrounding itself, and that is my corona radiata. So corona radiata is actually made of my granulosa cells. Some of the granulosa cell will come together and will get themselves attached to the zona pellucida. तो उसको हम बोलते हैं यहाँ पे as a corona radiata. So now you have a zona pellucida, you have a corona radiata are the two coverings you have surrounding the secondary oocyte. तो ये बात याद रख लेना. Please remember this thing, that surrounding your secondary oocyte, you can see these two coverings can be seen. First covering is my zona pellucida and the second covering is my corona radiata. क्या ये बात याद रहेगी आपको यहाँ पे? Will you able to remember this thing? अच्छा, thumbs up कर रहे हो?
सर आउटर कवरिंग पूछेंगे तो भाई क्या बोलना है?
हाँ, तो अगर see, they will ask you very precisely. If they ask you what is the outer covering of oocyte, primary oocyte or secondary oocyte, the outer covering of secondary oocyte, which is non-cellular, and that is my zona pellucida. क्योंकि zona pellucida एक cell नहीं है ना. Zona pellucida is actually a glycoprotein, right? So they will ask you very precisely that what is the outer layer of primary or secondary oocyte का outer layer क्या होगा, and that outer layer has to be acellular. Then that is my zona pellucida. If they ask you, they will ask you like this: what is a layer which is seen after zona pellucida surrounding the secondary oocyte? Then that is going to be my corona radiata, and corona radiata are actually cells. Corona radiata जो आपको यहाँ पे covering जो आपको दिख रही है ना, it is actually made up of the cells. So number of cells come together and they get attached to the zona pellucida. So that becomes my corona radiata. So ये बात याद रखना. So that should be clear to all of you. So Mantasha and Gayatri and Shikha and all of you should be clear with this. ठीक है? तो ये बात आपको यहाँ पे clear cut याद रखनी है. Now let's do one thing. तो इतना हमने यहाँ पे देख लिया. अब अगली चीज पता है यहाँ पे क्या होगी? The next thing I'll be finding here is ovulation will happen. Now my Graafian follicle will rupture. Graafian follicle मेरा rupture करेगा and the secondary oocyte is getting released. So now I'll just mention over here. Now the next thing which you can find happening over here is Graafian follicle is going to rupture. The Graafian follicle is going to rupture. Now, as soon as it ruptures, what has been observed? The ovulation will take place. As soon as the Graafian follicle ruptures, or I can say as soon as the Graafian follicle is going to rupture, what will happen? The secondary oocyte will get released. Your secondary oocyte is released. And as soon as the secondary oocyte is getting released, जैसे ही यहाँ आपने होते हुए देख लिया, this event is called as my ovulation. This event which has just taken place, this event over here is called as my ovulation. ये जो event मेरा यहाँ पे हुआ, इस event को मैं बोलता हूँ यहाँ पे as a ovulation. This event over here is called as your ovulation. So during ovulation, what will happen? The oocyte is released in the like secondary oocyte has been released, and do remember उसने अभी तक meiosis 2 complete नहीं किया है. याद रखना, secondary oocyte ने अभी तक आपका meiosis 2 complete नहीं किया है. मेरी बात समझ रहे हो ना आप लोग? I hope it's clear to all of you. उसने मियोसिस 2 वहां पे कंप्लीट नहीं किया, वो अटका हुआ है मेटाफेज 2 के अंदर. सो दैट शुड बी क्लियर टू यू कि मियोसिस 2 कंप्लीट करना बाकी है, वो करेगा, वो बाद में करेगा. बट इतना याद रखना, जब वो रिलीज कर रहा है ड्यूरिंग द ओव्यूलेशन, द सेकेंडरी oocyte is getting released. During ovulation, the secondary oocyte is getting released over here. Okay, so Graafian follicle is going to rupture and the secondary oocyte will get released. So that should be clear to all of you, right? तो इतना पहले हमने यहाँ पे देख लिया. Now let's do one thing because we have last 10 minutes. We have to solve some questions. I hope this is clear to all of you. यहाँ तक सबको कहानी क्लियर है या नहीं, बता दो सच्ची. हाँ, okay, ठीक है, all right. Now let me jump on to the questions over here. Let me, ये सब तो ठीक है, ये सब बाद में भी देख सकते हो आप लोग. Let me jump on the questions. Now bidder place of the sperm contains. Unmute करके आंसर दो सब लोग, unmute करके. क्या होगा इसका आंसर?
Mitochondria.
Right. The middle piece of the sperm is going to contain your mitochondria. Very good. बिल्कुल सही. ठीक है. अब ध्यान से देखना और इसका हैंड रेस लेके आंसर देना. How many sperms are formed from the secondary spermatocyte? How many sperms are formed from secondary spermatocyte? Secondary से पूछा है. Raise your hand and try to tell me the answer. बताइए गायत्री क्या होगा इसका आंसर?
4.
गायत्री is telling 4. ठीक है, बाकी लोग भी बताएंगे. तनिष्क क्या होगा इसका आंसर?
4.
And Shikha, what will be the answer over here?
So 4.
All of you have an incorrect answer. वे सबने बिना सोचे एक ने 4 बोल दिया, बाकी सबने भी यहाँ पे 4 बोल दिया. बिल्कुल आपके यहाँ पे इनकरेक्ट आंसर है. अब सोचो मेरी बात ध्यान से. सेकेंडरी स्पर्मेटोसाइट से आप स्टार्ट कर रहे हो. You are not starting from a primary spermatocyte. If you are starting from the secondary spermatocyte, then how many spermatid I'll be getting over there?
8.
अरे 8 कहाँ से होगा? अरे बाबा, समझो ने इस बात को ध्यान से. अगर आपका ये है मेरे यहाँ पे प्राइमरी स्पर्मेटोसाइट, assume that this is my primary spermatocyte, right? ये करता है मेरा यहाँ से, अगर मेरा यहाँ पे अगर होता है मेरा मियोसिस, मुझे क्या मिलता है? मुझे मिलता है मेरे यहाँ पे सेकेंडरी स्पर्मेटोसाइट. मिलता है नहीं मिलता?
Yes sir.
I get the secondary spermatocyte over here. अब तुम बताओ, जब ये मेरा सेकेंडरी स्पर्मेटोसाइट जब मेरा करेगा मेरा यहाँ पे मियोसिस कंप्लीट, जब यहाँ से मेरा मियोसिस किया जाएगा, यहाँ से जब आगे आप बढ़ाओगे स्टोरी को, तो कितने मुझे मिलेंगे मेरे यहाँ पे सेल?
2.
Will get 2 spermatid. So कैसे आपने बोल दिया 4 बिना सोचे हुए? You cannot do such mistakes. This is not acceptable by any means. So I am telling you, मैं underline कर रहा हूँ यहाँ पे कि it's a secondary spermatocyte. You have to be careful over there. It's not 4. The answer is going to be by 2 over here. So 2 is going to be the correct answer in this. Then, in humans, at the end of the first meiotic division, the male germ cell differentiates into. ध्यान से देखो, again, carefully look at this question. अगर आपने notes बनाया तो notes को देखो and then try to answer this. In humans, raise your hand. In humans, at the end of the first meiotic division, the male germ cell differentiates into. तनिष्क बताओ क्या होगा इसका आंसर?
Secondary spermatocyte.
तनिष्क के यहाँ पे सेकेंडरी स्पर्मेटोसाइट. बाकी लोग, गायत्री, what will be the answer over here? गायत्री,
Sir, I can't understand the questions.
what is happening, Gayatri?
Sir, I can't understand the questions.
Why it is so difficult to understand? See, over here, in humans, at the end of the first meiotic division, when the first meiosis happens, the male germ cell differentiate into. Now tell me something. The first meiotic division happens with whom? Spermatogonia या primary spermatocyte? किसके साथ होता है first meiotic division? Spermatogonia या primary spermatocyte? बोलो.
Primary
Primary
spermatocyte.
spermatocyte, right? So when this first meiotic division of primary spermatocyte will happen, then what we will get over there?
Secondary spermatocytes.
Secondary spermatocyte. So that is why option D becomes the correct answer.
Oh, thank you.
You need to understand the question. You have to understand the question, then you have to answer them. You cannot jump on the question directly. Sertoli cells are regulated by pituitary hormone known as. Raise your hand to answer this. Mantasha, Shikha, hand raise करो. बताया था आज के क्लास में हमने कवर किया है. Sertoli cells are regulated by pituitary hormone known as. Who is going to go and tell the Sertoli cell to make something? Tell us, Shikha.
Sir, FSH.
Mantasha, क्या लग रहा है आपको इसका आंसर?
Sir, FSH.
Right. FSH is the one which will go and tell the Sertoli cell to make something, to trigger and to make something over there. So yes, that is my FSH over there. Correct answer. Now again, read the question very carefully. The correct sequence of spermatogenetic stages leading to formation of sperm in the mature human testes. So tell me the correct stages over there. So take your time, take your 30 seconds and tell me which is the correct stage over here. Again, a very simple question. You just have to pay attention. तनिष्क क्या होगा इसका आंसर?
Sir, option A.
तनिष्क कह रहा है यहाँ पे ऑप्शन है. बाकी लोग मंताशा, शिखा एंड गायत्री. शिखा क्या लग रहा है?
So option A.
Right, option A becomes the correct answer. Spermatogonia will make the spermatocyte, then you'll have the spermatid will form, and then the sperms will be formed. So that is why option A over here becomes my correct answer. Again, read the question very carefully. Which of the following statement about human sperm is going to be correct? So you have this certain statements over here. Read them carefully and then try to tell me the correct answer. You have to read by yourself. Gayatri, tell us.
Sir, option A.
Okay, that is saying option A. Other people. Tanishk.
Sir, option B.
Option B. शिखा.
So option B.
मंताशा बताइए बेटा, आप भी बताइए. जब तक क्वेश्चन के साथ एंगेज करेंगे नहीं तब तक ग्रिप कैसे बनेगा टॉपिक के ऊपर? बोलिए क्या होगा? गलत ही आंसर होगा ना, ज्यादा से ज्यादा कुछ नहीं होगा. फिर क्या होगा इसका आंसर?
Sir, option A.
कभी भी आंसर देने से हेजिटेट मत करिए. कुछ नहीं होना, ज्यादा से ज्यादा गलत आंसर आने वाला है और कुछ नहीं होगा. उससे ज्यादा ये होगा कि सर डांट लगाएंगे, उससे भी ज्यादा कुछ नहीं होगा. और सिंपल सी बात है, डांट मैं ही लगाऊंगा, पड़ोसी नहीं लगाने वाले आपको. राइट, दैट इज समथिंग आई एम सपोज टू डू. ठीक है, नहीं होता ना किसी के घर में कि बाजू वाले अंकल गुस्से में आके बोलते हैं अरे क्या यार तनिष्क, यू डोंट नो स्पर्मेटोजेनेसिस, व्हाट इज हैपनिंग, हाउ दिस विल हैपन. नहीं होता ना तुम्हारे साथ कभी ऐसा. राइट, टीचर ही वो तुम्हारे साथ करेगा. सो प्लीज फील फ्री, आंसर योर क्वेश्चन ओवर हियर. राइट, गलत तो गलत, कोई बात नहीं, तभी एंगेज करोगे तभी सीखोगे. अब सुनो ध्यान से. ऑप्शन है यहाँ पे क्या कह रहा है? एक्रोसोम हैज अ कॉनिकल पॉइंटेड स्ट्रक्चर used in the piercing and penetrating the egg resulting the fertilization. ठीक है, ये लोग कहेंगे कॉनिकल स्ट्रक्चर होता है जो हेल्प करता है पियर्स करने में एग को ड्यूरिंग द फर्टिलाइजेशन. ठीक है. स्टेटमेंट बी यहाँ पे क्या करा है? द स्पर्म लाइसिन मतलब एंजाइम्स. ये मैंने आपको लिखा था वहां पे, याद करो मैंने लिखा था लाइसिन्स. स्पर्म लाइसिन मतलब द एंजाइम्स. द स्पर्म लाइसिन इन द एक्रोसोम dissolve the egg envelope facilitating the fertilization. अब आप सोच के बताओ ए और बी में से ज्यादा सही कौन सा है यहाँ पे? ऑप्शन बी. ऑप्शन बी आपका यहाँ पे ज्यादा सही, बिकॉज यहाँ पे स्पेसिफिक मेंशन किया है दैट यू हैव अ स्पर्म लाइसिन, यू हैव दोस बैग ऑफ एंजाइम्स इन द एक्रोसोम्स, एंड दैट्स ट्रू. इन एंजाइम्स की वजह से आप एनवेलप को ब्रेक डाउन करके आप अंदर जा पाते हो. ऑप्शन ए पता है क्या लग रहा है? ऑप्शन ए में क्योंकि उन्होंने एंजाइम का मेंशन नहीं किया, इट द स्टेटमेंट सेज दैट इट हैज अ पॉइंटेड एंड. वो इतना पॉइंटेड हो रहा है कि वो पियर्स कर पाता है, बट वो बात सही नहीं है. वो पियर्स इसलिए कर पाता है बिकॉज इट हैज अ एंजाइम ओवर देयर, इट हैज अ स्पर्म लाइसिन ओवर देयर, and because of the sperm lysine you are able to enter inside the egg. So that is why option B over here is going to be a correct answer. क्या ये बात आप सबको यहाँ पे समझ आ गई? अच्छा समझ आ गई, ठीक है. चलो आगे बढ़ते हैं. Again, arrange them carefully. NEET 2013 का क्वेश्चन है, ध्यान से देखो and raise your hand. Which is the correct sequence of the sperm formation? Mantasha, tell us.
So option B.
Right. And I hope all of you will agree with this. Option B over here becomes my correct answer. Spermatogonia, spermatocyte, spermatid, and spermatozoa. Okay. Which of the following cell during gametogenesis is normally diploid? इनमें से कौन सा सेल आपका normally diploid होता है during gametogenesis? Take your time and then tell me the answer. चलिए चलिए हैंड रेस करिए फटाफट. तनिष्क ने हैंड रेस कर दिया, बाकी लोग भी हैंड रेस करिए. चलिए जल्दी. तनिष्क क्या होगा इसका आंसर?
ऑप्शन ए.
मंताशा क्या लग रहा है?
Sir, option A.
शिखा.
So option A.
And Gayatri also. What will be the answer, Gayatri?
Sir, option is.
राइट, एंड दैट इज माय डिप्लॉयड स्पर्मेटोगोनिया. बिल्कुल सही. चलो ध्यान से देखो, बहुत ही अच्छा क्वेश्चन है. द डिफरेंस बिटवीन स्पर्मियोजेनेसिस एंड स्पर्मिएशन. याद करो दोनों को मैंने क्या समझाया था. व्हाट वाज माय स्पर्मियोजेनेसिस एंड व्हाट वाज माय स्पर्मिएशन. सो रीड this thing or remember this thing whatever we have done in the starting of the class and tell me the correct answer. The difference between spermiogenesis and spermiation is. Now read all the statement carefully. लो अपना 1 मिनट लो यहाँ पे and then try to answer this. Gayatri, tell us.
Sir, option D.
Gayatri is telling us option D. बाकियों का भी हम लोग आंसर लेंगे यहाँ पे. तनिष्क क्या लग रहा है?
ऑप्शन डी.
तनिष्क भी कह रहा है ऑप्शन डी यहाँ पे. शिखा.
ऑप्शन डी.
एंड मंताशा.
सर ऑप्शन डी.
पक्का 100%.
यस सर.
This is going to be your option D. बिल्कुल सही. सबका आंसर यहाँ पे सही है. In spermogenesis, spermatozoa are formed, right? While in spermiation, spermatozoa are released from the Sertoli cell into the cavity of my seminiferous tubule. So उसे वहां से हम रिलीज करके उसे आगे भेजते हैं. So that is something we'll find happening in my option D. So बिल्कुल सही, आपने सही डेफिनेशन पकड़ी है spermiogenesis की और spermiation की. This should be clear to all of you. Question number 7. The testes are situated dash in the abdominal cavity within the pouch called. So again, शिखा बताओ इसका क्या आंसर होगा?
सर ऑप्शन बी.
Option B, outside and in the scrotum. Yes, it is outside the abdominal cavity and in the scrotum. So then मुझे बता दो क्वेश्चन नंबर 8 का आंसर क्या होगा यहाँ पे. कौन बताएगा इसका 8 का आंसर? हैंड रेज करना पड़ेगा 8 का आंसर. 8 के आंसर के लिए हैंड रेज करेंगे. तनिष्क चलो बता दो क्या होगा 8 का आंसर.
ऑप्शन बी.
Right. Option B over here becomes my correct answer. So, which of the following is a primary sex organ in the females? A, B, C, D. Shikha. So much time. Shikha, बताइए.
सर ऑप्शन ए.
प्राइमरी फीमेल सेक्स ऑर्गन ऑप्शन ए. शिखा, घर पे फोन आएगा अभी, सीधे घर पे फोन आएगा अब तो.
ऑप्शन बी सर.
का टीवी बंद कराओ देखने का. टीवी देखते हो आप? नहीं देखते हैं. मोबाइल ले लो इनसे, पढ़ाई नहीं हो रही. ऑप्शन बी बेटा, ऑप्शन बी ओवरीज आएगा इसका आंसर. ऐसे नहीं गलत कर सकते, राइट? यूट्रस इज नॉट गोइंग टू बी द आंसर. यूट्रस इज समथिंग वेयर द इम्प्लांटेशन विल टेक्स प्लेस ओवर देयर. Okay, now think wisely and try to tell me the answer. Which of the following is not a accessory male duct in context of male reproductive system? Which of the following is not a male accessory duct? कौन सा इनमें से मेल accessory duct नहीं है? बताइए. हैंड रेज करना पड़ेगा. अरे बड़ा आसान सा सवाल है, इतना क्यों टेंशन ले रहे हो आप लोग? आसान सा सवाल है ये. इतने क्वेश्चन को देख के इतना टाइम लगेगा तो कैसे चलेगा? आवाज आ रही मेरी या सब लोग चले गए फोन रख के? सब लोग बालकनी में फोन रख के चले गए. आ रही है आवाज तो क्यों इतना, क्या किसका वेट कर रहे हो? बताओ तनिष्क क्या होगा इसका आंसर?
बी.
टेस्टिस सही आंसर है यहाँ पे. ये आपका एस्ट्री डक्ट नहीं है, बाकी सारे आपके एस्ट्री डक्ट है. वास डिफरेंस आपका हो गया, एपिडिडाइमिस आपका हो गया, रेडियस हो गया. दे आर ऑल माय एस्ट्री डक्ट्स. दिस इज माय प्राइमरी सेक्स ऑर्गन, दैट इज माय टेस्टिस. ऑप्शन बी बिकम्स द करेक्ट आंसर इन दिस. इतना सोचना पड़ता है आपको इसको आंसर देने के लिए. ठीक है. नाउ व्हाट आई डू ना, यू कैन हैव अ लुक एट दिस क्वेश्चन बाय योरसेल्फ. बाकी 4 क्वेश्चन एक बार घर जाके एक बार खुद से देखो. बट उससे भी ज्यादा इंपॉर्टेंट है कि आज हमने किया है oogenesis और spermatogenesis कंप्लीट. दोनों चीजें आपको एनसीईआरटी से पढ़ के आनी है. अब एनसीईआरटी में कुछ चीजें थोड़ी सी अलग दी हुई है. जैसे एनसीईआरटी में लिखा हुआ है कि आपका ज़ूना पेलुसिडा बनता है व्हेन यू फॉर्म द सेकेंडरी स्पर्मेटोसाइट. सॉरी, सेकेंडरी oocyte बनने के बाद यू गेट टू सी अ ज़ूना पेलुसिडा. बट एक्चुअली वो पहले से ही बना हुआ होता है, राइट, फ्रॉम द प्राइमरी oocyte. बट उसमें वहां पे लिखा हुआ है तो आप वो फॉलो कर सकते हो, वो फॉलो करोगे, राइट? सो बट एक बार फिर एनसीईआरटी से पढ़ो, जो भी हमने जहां तक हमने देखा है उससे पढ़ो एक बार, एंड प्लीज रीच आउट टू मी अगर कुछ समझ नहीं आता है तो, ओके। एंड आई वुड स्ट्रांगली रिकमेंड कि जो मैंने यहाँ पे ड्रॉ किया हुआ है, लिखा है, उसे एक बार खुद से लिख लेना, उसे एक बार खुद से ड्रॉ कर लो, लिख लो एंड रीड द एनसीईआरटी। ये क्लियर रखना ताकि इसमें से कितना भी कोई घुमा के क्वेश्चन पूछ ले आपको आना चाहिए, ठीक है। सो ये चीजें कर लो, राइट। आई बी शेयरिंग विद यू, आई वोंट शेयर विद यू द असाइनमेंट राइट नाउ फॉर दिस। हम लोग जो क्वेश्चन दे रहे हैं वही हम अभी कर रहे हैं सॉल्व इसके अंदर। आई विल शेयर यू द असाइनमेंट फॉर फ्लावरिंग प्लांट्स को लेके एक और असाइनमेंट शेयर करूंगा, वो करना सॉल्व, ठीक है। सो क्विकली सॉल्व करो, ऐसा नहीं कि आज दिया फिर 2 दिन बाद आ रहा है, आज अगर मिल जाए तो कल सुबह तक सॉल्व करके वापस करो, इट वोंट हैव मोर क्वेश्चंस, राइट। सो डू दैट, ठीक है। सो दैट इज समथिंग वी हैव डन, आई होप इट्स क्लियर टू ऑल ऑफ यू व्हाट वी हैव डन आज के क्लास के अंदर, एंड ठीक है, विल कैच यू अगेन आफ्टर सम टाइम। सो इफ यू हैव एनी क्वेश्चन देन प्लीज गो अहेड एंड आस्क अ क्वेश्चन। जिनके डाउट्स आए थे असाइनमेंट को लेके वो अपने डाउट्स क्लियर कर सकते हैं